Okay, so I guess we can get started, right?
Yeah.
Well, I would like to thank everyone for coming to the workshop personally. Also, I think it's a bit practical that we don't have a microphone anymore this year. It felt a bit awkward last year, but it's nicer with a webcam. So, okay, I'm going to talk about Markov models actually again. So, thank you very much actually to Guillermo for the nice presentation on Markov models. So I might repeat some things, but I think it's good in any case to understand it better. And I'm going to repeat them in a different manner, so from a different perspective. So essentially what I'm going to talk about is a bit about the general MSM pipeline, how to build an MSM. Then I'm going to talk about some Markov linearity tests, which you will have to know, at least in my Timescales one. Chapman-Kolmogorov maybe not so much, but it's nice to understand. Then we're going to talk about discretization problems, which Guillermo also mentioned. Macrostate lumping, so how to go from microstates to macrostates. And lastly, I'm going to talk about the implementation of Markov-state models in HTMD. And then I will also show you in code in the notebooks how we do all of the stuff in HTMD. Okay, so when we typically have a large set of simulations, in our case, they normally consist of hundreds or thousands of short simulations. So the question is, how do you go from those short simulations to reconstructing something like the energy surface of the protein, understanding the kinetics between the states, and all of the stuff? I actually also found the source of this image, which we are putting everywhere.
Is that the source?
Yeah, that's the source. It's from Vivian. That's some.
I think Frank gave this as a. So we tasked somebody, like some artist.
Ah, it was.
Yeah,
okay, it was tasked by Frank. Okay. Yeah, I was wondering because everyone was using it everywhere.
Yeah.
Okay, so one good way of reconstructing essentially this the kinetics is a Markov-state model, and they have had lots of success in the recent years. So we start with our set of simulations. We project them on some lower dimensional space, in this case like a two-dimensional space as an example. So you can imagine that each dot here corresponds to conformation of the protein. Then we can cluster this space into a set of states, which we're going to use for the Markov model. And then we're going to count transitions between those states from the state itself. And that's how we build the Markov model. And from the Markov model, we can calculate then all of the kinetics that we are interested in the metastable states, etc. So let's go into a bit more detail. So why do we do the dimensionality reduction? I mentioned that we do a dimensionality reduction. Practically, if we have our data on a super low dimension where everything is separated nicely into two clusters, let's say, and we add another dimension to our data, then our data becomes spread out in space. And now if you do a clustering, you might get a totally different clustering of your states at this point. So what the curse of dimensionality says is that the more dimensions you have, the more spread out your data becomes, the more hard it becomes for clustering methods to detect your clusters. So we want essentially to have a low dimensional representation of our data. So because the simulations are very high dimensional, they're 6 times the number of atoms. So if we have 500,000 atoms, it's an insanely large dimensional space. So even if we throw away the velocities, we are at half that space. So we do some projections, like the distances between carbon alpha atoms of the protein, for example. And this end up being something like 600 dimensions, maybe 1,000 dimensions, which can still be very high dimensional. So sometimes we need the other methods to go even lower, like TICA, for example, that Guillermo presented before. So in more detail, the Markov model pipeline, as I said, we start with a projection. So we want to reduce the dimensionality for data. For example, in protein ligand binding, you might look at the contacts of the ligand with the protein, like here. So you're looking at the carbon alphas with which the ligand comes into contact. So it's closer than 8 angstroms, for example. And this is a quite low dimensional representation because it has the same number of dimensions as the residues in your protein. So if you have a 300 residue protein, it's a 300-dimensional vector which represents each one of your poses. Okay, so as I said, yeah, every simulation frame that we have will get projected onto a one-dimensional vector. Sorry, no, a vector of multiple dimensions, depending how many we chose. And then we're going to cluster them depending on how similar they are, the conformations. So if you can imagine that this here is like Joao showed you yesterday, that you can use this metric RMSD or the metric distance. So this produces a two-dimensional matrix in which the columns correspond to your dimensions and the rows correspond to the frames of the simulation. So this is one simulation. Each row is a frame of the simulation, a time frame, and each column is a dimension. So once we cluster this, we can project each one of the frames to a specific state. So once we've done the clustering, so you can imagine that this conformation corresponds to this state. So if we have a trajectory which goes like this through the states, you can imagine that we can represent it as a trajectory just of labels going from each state to another state. And then we can calculate the count matrix as Guillermo said. So we can do simple counting. So we go from A to A, so we put one count here. We go from A to B, we put one count here. From B to B, we put one count here, etc. So we do the counting of our transitions. And as I mentioned, you can do this counting with different lag times. So depending on what lag time you choose, you get a different count matrix. So if we choose a lag time of 1, then you count the transitions of each frame to the next frame. If you choose a lag time of 2, then you count the transition from each frame to the 2 frames afterwards. So from A to B in this case, from A to B again, and from A to A. So I'm going to talk about later what the effects are of this lag time. And then as we said, we calculate a transition matrix, which you can do the stupid way from the count matrix by just dividing by the sum of the rows, as mentioned, or you can do something a bit fancier typically, but you don't need to care about that much. So, okay, once we have a transition probability matrix, we can calculate anything that we want from the Markov model, mean first passage times to go from one state to another, the most metastable states, the kinetics, and more. So as I said, we have our simulations, we project them, cluster them, count matrix, transition matrix, and we're done. So let's talk about Markovian tests. No one guarantees that our dynamics are Markovian in the discretized space that we have produced. So there are two main tests that we typically do: the implied timescale plot, which hope I didn't break. Okay. The implied timescale plot and the Chapman-Kolmogorov test. So to introduce the implied timescale plot, I'm going to go a bit into the theory part, the eigenvectors and the eigenvalues. So if we have this 4-well potential here, and we build a Markov model out of it, you can calculate the eigenvectors and the eigenvalues of that transition matrix. So if we look here at the eigenvectors, the first eigenvector corresponds perfectly to the equilibrium population of those states. Okay, so yeah. So once we look at the rest of the eigenvectors, we can normalize them by the first one, and then we get this here. Actually, Guillermo, if I say something wrong, correct me, because we get these values here. So okay, the first one will be 1 because we normalized the itself. But if you look at the next one, at the second eigenvector, you see that some states are on the negative side and some are on the positive. And the point where the transition goes from negative to positive is where your largest barrier is in your system. So this one tells you that the largest barrier is between states A and B and C and D, which is true, this barrier. That's exactly what it shows here. The next one tells you that the second largest barrier is between A and B. That's why it goes from positive to negative. And the third one tells you between C and D. So this is perfect. We can find essentially where our large barriers are. So each eigenvector corresponds to a slow process in the system. And the eigenvalues, on the other hand, the first eigenvalue will be 1 always in our cases. So we can ignore that one because it corresponds to the equilibrium population. But the rest of them, they correspond to the time scales of the slow processes. So the second time scale, so the second eigenvalue will correspond to the time scale of the second process of going from AB to CB. Actually, if you have any questions or you haven't understood something, just let me. And the next, okay, so they all correspond to the timescales of those processes. So now we can look at the implied timescale plot, which I hope it makes a bit more sense now. So we take the eigenvalues. So sorry, we build a Markov model at different lag times here. For example, at this lag time here. Yeah. Yeah.
Yeah. If these two eigenvalues, the first two are degenerate, then the first two eigenvectors can also be mixed in many different ways. How would you interpret that?
I don't know. I mean,
yeah. So essentially, if the only way that you're going to get two eigenvalues with the value 1 out of a row or column stochastic matrix is if the matrix itself decomposes into two classes.
Yeah.
Which means that in your system there's either some irreversibility, so you have part of the space where some process happens that as soon as it goes to some other part, it never goes back. Or you have completely disconnected sets.
Yeah.
And thus, each set gives you an eigenvalue 1, because each set has its own stationary distribution, the first eigenvector there. And that degeneracy that you're speaking is that any combination of these two stationary distributions are a stationary, any combination of the first two are still an eigenvalue of the matrix.
Yeah, as long as it's connected, all of it, and it's embodied.
Well, yeah.
That you can get from each state. Yeah.
It cannot really be reversible without being embodied. So.
Yeah, then it has a single eigenvalue, which is always 1. That's the point. Which corresponds to the equilibrium probability of that model. Okay, so yeah. You can choose different lag times to create your Markov model, and so we do this for multiple lag times, and we calculate the eigenvalues, and then we just connect them with lines here to see at which point they start to become flat. So typically, when we choose a lag time for our so on the X we have the lag times, and on the Y we have the timescales or the eigenvalues. So we typically choose a lag time for our model when the timescales start to become flat. In this case, you could choose something like here, for example. In this timescale here on the right, they become flat a bit later, but they're still quite flat enough, I would say. So the last thing that I want to point out is the thing called the spectral gap, so the gap between the timescales. In this case here, you can see that we have one really slow process and maybe another 3 slow processes, and the rest of them are very, very fast. So that means that we have either one slow, you can look at either one slow process or 4 slow processes. And as I showed, each slow process splits your states into one more state. So if you look only at the first slow process, you have 2 states that you're looking at. And the more you add more states. So if you want to analyze the first 4 slow processes, you're going to need 5 states. Here it's a bit not so clear, but I would say something like 2 or 3 slow processes, probably, maybe even 4, but it's a bit debatable. So it's not such a super clear thing sometimes how to choose how many states you want. So the Chapman-Kolmogorov test, which I'm going to talk about. So the Markov models, they have the slides that I'm using are most of them from Frank's group, essentially from Jan Henry. So the Chapman-Kolmogorov test, essentially when we have a Markov model, we can predict steps in the future. So what we want to do is we want to see if our model corresponds to what we saw in our simulations. And it's exactly what we see here. So we say that if we start in state A, what's the probability of staying in state A after this amount of time? And then we check how often, what our simulations tell us, which are these error bars, and then we see what our model tells us. And as you can see here, the model agrees quite well. It's within the error of the simulations. Exactly. So we want to see if our model matches our simulations, essentially. But we can look at another thing here. So if we build a model with a lag time of 100, we can see that it doesn't really match our simulations, which are these ones here, the black ones with the error bars. If we increase the lag time to 500, then it's starting to match a bit more. If we increase it to 2000, then it matches nearly perfect. Also here on the right, you can see that if we increase the number of states, then it starts to match even better the stuff that we see in the simulations. So essentially what this tells us is that we have 2 ways of solving the problem, which is one is to increase the lag time, but then we lose statistics if we increase the lag time too much. The second solution is to improve the clustering, which is better. And for that we can use some clever methods like TICA, which are going to improve our clustering. So this brings me to the discussion of discretization. So what is the problem essentially with the short lifetimes? And why do we get this fast kinetics also? So practically when you have your continuous dynamics, like in this example here, in this column, we have this double well potential. All of our population starts, for example, in state A. As time goes by, population starts flowing from state A into state B, as you can see here. And after the next step, we have like 0.088. Now, if you do the same thing, but with discrete, okay, I wrote this wrong, it's discrete TE dynamics, you can see that, okay, it works fine up to this step, but here they actually disagree on the populations of the states. The reason for this is that when you're propagating your probabilities with the Markov model, you're missing information about stuff that's between your two states. You don't have any information about the populations there. So populations that would be somewhere here between those states in the continuous dynamics, they're either thrown into the A basin or into the B state. So every time that we do a step in the future with our Markov model, we're making an approximation error. So the larger the lag time is, the less times we're going to make that approximation error. So the more similar it's going to look to the continuous dynamics in that case. So what's the best discretization? Okay, let's look at this double well potential here, the equilibrium population of that double well potential. So if we look at the second eigenvector, which is this black line here, the black curve here, if we split the states directly in the middle, and we have one state here, one state here, then the approximation error of our eigenvector will be this red area here, because we approximate the eigenvector as a step function, like this value here and that value there. If we move the barrier to the left, then it's going to be a horrible discretization of our space, and we're going to have a horrible error here, a very large error of the approximation. If we add just lots of clusters uniformly in our space, we're going to have a relatively small error. But the smallest error that you can get is by putting as many clusters as you can on the transition regions. And exactly the same thing you can see here. Essentially, you can split it perfectly on the barriers into 3 states, but then you have quite some error. You can split it into thousands of states, then you have a small error. Or you can split it intelligently on the barriers, and then you have an even smaller error of your discretization. So essentially, we want to put as many clusters as we can on the barriers. That's the most intelligent thing to do when building Markov models. So this brings me to TICA. So everyone knows PCA, I guess, which projects your data on the dimensions of largest variance. TICA tries to project your data on the dimensions of your slowest reaction coordinates. So in this case, like in this beautiful example of this two-well potential, PCA would project on this dimension, while TICA would project on the orthogonal to that one. So, which would be perfect because it projects exactly on the slow transition, which is to go from this basin to this one here. So by projecting on this, it allows us to put lots of clusters on the transition regions and thus improve the clustering. Okay, so about macrostates, if you have any, well, yeah, as I said, if you have any other questions, please ask me about it. So how about the human interpretation? So as we said, okay, I also forgot to mention that, okay, if we cluster a space into so many clusters that we improve the discretization, then you have another problem, that you have a statistical problem again. You have like 5 frames left in one conformation and 2 in the other one, so what statistics are you going to count between them, between those states? So, however, we typically build models with thousands of states. This makes it very complicated to interpret. So what are you going to look at? Are you going to look at all of the 1,000 states and then look at the kinetics between those? So we need to reduce the number of those states with what we can call like a lumping. So you want to group together your microstates, which are the small samples here, kinetically together into some macrostates, which interconnect very fastly, but are separated kinetically. That's the ideal thing. So how we do it? We typically do it, in our case, we do it with PCCA because it's a really fast method. And there are better methods, like HMMs, for example, can do a better separation, better lumping. But they take very long, like half an hour or 1 hour or something. PCCA takes a few seconds. So we have that as default in HTMD at the moment. So what it does, as I explained to you before, it looks at the eigenvectors and then just uses the eigenvectors to split your states, your space into the most metastable states, like first A and D. If you ask for only 2 states, it's going to give you A, B, and C, D, sorry. If you ask for 3 states, then it's going to give you A, B, and C, D. So it uses the barriers and the eigenvector structure to give you a simplified model. So we can simplify a 1000-state model essentially into a 3-state model, which is really nice, so that we can interpret it better. Okay, so let's talk about the implementation in HTMD of Markov state models. So I wanted to mention one thing that Johnny also mentioned yesterday, which is that we consider kind of our folders as so each simulation needs to be in a separate folder, first of all, and we kind of consider them as units of execution. So we can imagine that we have a folder called 1, which has all of the input files that were used to run your simulation. It might even contain your trajectory of the simulation if you want to. And the name of the folder is the name of that your simulation is going to have. So typically what we want, well, not typically, what I usually do is that I separate my inputs of the simulation from the trajectories, which I put into folder data. Practically, if you want, you can put your simulations into the same folders as the input files where you started the trajectories from. So otherwise, we need to do some matching of these folders, typically by the name of the folder. So if this folder here is called 6, we're going to match it with the folder 6 in data, where the trajectory of the simulation is stored. So this is done by a function that's called simlist, which I'm going to show you later in the more practical part, which tries to link the simulations to the PDB structures which are found in those folders, to the structural information of the simulation. Okay, so the main components then of HTMD, to explain to you what the workflow is in HTMD when we try to build a Markov model. We first create a simlist, as I said, so we link our simulations with some structural information so that we can do projections. Like Joao showed you yesterday, when we want to do a projection like RMSD or something, we need to load a molecule, which contains the PDB structure or any structural information, the PSF if you want, and the trajectory, an XTC file, for example, depending on what formats you use. So this links the simulations with some structural information, as I showed you before. Next, we have our projections, like metric distance, metric RMSD, metric plume that were presented yesterday very nicely. So we take the sim list and the projection, or multiple projections if we want, and we pass them to a class called metric. And this metric class then produces an object of another class called metric data, which is your projected data. So then we have the options of either clustering directly this metric data, but it's quite high dimensional, as I said. So if we do like carbon alpha distances of a protein folding, you might have like 1,000 dimensions, which is really high for clustering. It's not going to work really well. So you can either cluster, or you can pass it through TICA, which produces another metric data object, which you can cluster now because it's in a lower dimensional space. And whichever you choose to pass essentially to the model, you choose if you want to use the unprojected data or the projected data by TICA, and you pass it to a class called model, which calculates your Markov model nicely, calculating the transitions between the states. And all of this gets fed into another class called kinetics, which calculates your kinetics. So essentially, you need to know only 4 to 5 classes, which are metric data, which is produced by metric, TICA, which you can use or you can choose not to use, but you should use. Then the model class and the kinetics class, and the simlist function. That's everything we're going to look at now in the Jupyter notebook. Okay, so let's go to our notebook, which is located in the folder 08 MSM analysis. This one here. We won't be able to execute everything here because some stuff took like 2 or 3 minutes, and I didn't want to waste too much time with that. Like TICA takes a bit of a while, also plotting timescales. So Some cells might not execute. I will just tell you which ones you can execute. So we're going to analyze the folding of billion, which is this protein here that you can see. So first we're going to create a list of our simulations with the SIMList function. Then we're going to project our simulations. We're going to project them again using TICA. We're going to cluster the TICA projected data. We're going to build the Markov model and calculate the kinetics. This is the pipeline that I was talking to you about. So here we create the SIMList. And the way that we do it is with this function. We provided a list of data folders where your trajectories are located. Each folder corresponds to one simulation. And then you pass it another directory, which is this input directory, which contains the structured PDB files, for example. Internally, this function is going to link the folders based on their names that they have here. So if the folder is called 1 here, it's going to look for a folder called 1 here and use the PDB structure from that folder. And it's going to produce a sims variable, which is just a Python list. So we cannot execute this because I haven't shipped all of the data, but I'm going to. Yeah?
Just quickly, so does it know what because you mentioned that your folder is kind of like your unit of simulation,
yeah.
Assuming the folder files, but this blob is just producing the list of folders?
Yeah, it just produces a list of folders and then
the intelligence to just pick up the trajectory files?
Yeah, it looks internally to find any recognized trajectory type. Although it's going to stick to the first one that it finds. So if you have ecstasies and DCDs, it's not going to merge them together. It's going to pick, for example, only the ecstasies. So take care of it without like having different trajectory types.
It's going to pick one file per folder?
No. You can have multiple. So if your trajectory is split into pieces, just put them all in the same folder and take care that the naming is correct, so that it's a natural naming like 0, 1, 2, 3, etc. And then it's going to sort them correct and load them all together into the same trajectory. So filtering trajectories, we often remove waters in our simulation because we don't need them in the analysis. Speeds up everything that we do, and we typically, when we have like multiple different starting structures in our simulations, they differ in the number of waters. So it's practical to remove the waters because then we have a unique structure for all of your trajectories, which we can use for visualization also. So here you provide just where you want to store your filtered trajectories. You provide a selection, what do you want to keep, so not water, everything that's not water, and produce your new sim list here.
You also go from terabytes to gigabytes. So.
Yeah, once you remove the waters, it's quite important. Okay, so what happens though if you have folders with the same simulation names, which happens from time to time, especially if you do adaptive sampling, because adaptive has like a specific naming scheme. If you do different runs of adaptive sampling, like total independent, you might end up wanting to join all of this information in a single model, but they have the same name. So what do? First of all, let's list the folders that we have. We have 3 folders here of adaptive sampling of the same protein. So here then, so you can start executing actually from here on, if you want. So we create a sim, so we just loop over the folders and we create a sim list for each one of them, and then we merge them into a unique sim list called sims. And as you can see here in the first folder, there's 720, 710, 708 simulations, and they all get joined into a sim list of 2,100 something simulations. So now we can do our projection. As I said, we have our metric class, to which we pass our sims, which is our simulation list. And then we set a projection that we want to do with that class. In this case, I chose to do the distances between the carbon alpha atoms of the protein. Actually, not the distances, the contacts. The contacts between the carbon alpha atoms of the protein. And then you call the metric.project, which projects your simulations and produces this metric data object that I talked to you about before. And as you can see, it's quite fast. So for 2,100 simulations, it does it in like 30 seconds, which is good, especially on those machines and EC2 machines. Yeah. It also throws you a warning very often because simulations are supposed, depending on your software, they are supposed to contain the frame step. So the frame step is the time which passes between each frame of the simulation, or your sample time or something you might call it. So it complains that, okay, there were multiple different frame steps read, but it's picking 0.1 nanosecond as the statistical mode. If it looks wrong, you can change it. So you might want to set it like this: data.fstep is equal to 0.1, which units are in nanoseconds. we didn't need to do it here because it actually gets the correct time step, but frame step, but it's good to know. So then we can plot the sizes of the trajectories if we want, to the lengths of our trajectories. Yeah, produce this plot. Okay, this is a bar plot, but because we have like 2,000 bars, they get kind of joined into a single thing looking like a box. So you might think here that all of our simulations are actually of 500 frames, but you are going to be wrong. Because sometimes we have some corruptions in our simulations, which end up with some simulations which are shorter than they should be. The simulations are not just shorter, like we lost the last frames, they're shorter and they are pretty bad, they're corrupted. So if you build a Markov model with those, you're gonna make a really bad model. So we'll just throw them out.
We run this on computer, which are totally volatile. They switch off and on.
Yeah, on GPU grids. Yeah.
So ACEMD has a perfect checkpointing situation. So if indeed something breaks, the power is on, and when it starts again, it will restart, see what it is, and append the XTC. Now, sometimes this procedure is not clean, because it depends. Maybe it was writing while the writer decided to switch off the machine. So sometimes we have this situation. In a cluster, you shouldn't have this problem.
Yeah, okay. But actually, this function is still useful in many cases if you want to drop a specific some trajectories. So, for example, you can provide some limits. You can tell it I only want to keep trajectories which are longer than 50 nanoseconds and shorter than 100 nanoseconds. Or you can tell it that I want to keep only trajectories which are a multiple of 50 nanoseconds. So it actually has some nice arguments if you want to drop some of your simulations. Now we can go on to TICA. I'm not going to execute this because it takes around 3 minutes. So as we said, the distance, so if you calculate the self-distances of the protein, they're going to be like n times n minus 1, n being the number of residues, divided by 2 dimensions, which is quite high dimensional. We want to project it with TICA. So TICA has 2 arguments. It has a TICA lag time, which is not related to the Markov model lag time, so don't confuse it with that one. The correct lag time, the best lag time is a bit debatable. We typically put something like 20 nanoseconds, but it's, well, in some cases even shorter. I don't know if you have any good suggestion about TICA lag times. I haven't heard any good theories until now. But.
I mean, we, periodically having this conversation every time we meet, it's a little bit like the answer I gave before. If you can observe a converged timescale plot for the TICA, then you should go with that timescale. If you cannot, then you just fix the lag time and say, I am ignoring everything that's faster than 20 nanoseconds.
Yeah.
In many cases, the MSM is very robust with respect to this choice because you're only using TICA to guide you to the slow space. So if you cluster in the first 10 ticks, but actually the slow things are happening only in the first 5, the order of these 5 or of these 10 doesn't really play a role. So, but yeah, to be short, between several nanoseconds and tens of nanoseconds is usually what we use.
Yeah. And the next argument is the number of dimensions.
We did see significant difference in the output if you go from 2 nano to 10 nano.
Yeah.
10 nano a lot better than 2 nano.
Okay, well, we need to check.
I don't know why. I don't know if it's generic.
Well, also there is an issue with high dimensions in TICA, but that's another subject we can talk about another time. In any case, typically we project on 3 or 5 dimensions with the project on TICA. Yeah, keep below 5 dimensions with TICA and you're gonna be fine. We cannot call this commands because I skipped the TICA part, but I'm gonna tell you when we can go on. So here you just, this metric data object, you cluster it. Here we cluster it with mini batch k-means, which is an sklearn clustering method. So you can use, I think, any kind of sklearn clustering method. We need to test it for promising much, but we typically use this one. It works very well for our data. We pick something like between 500 and 1,000 clusters to produce. And then you create your model by passing the cluster, the metric data object, to the model class. Then you can call the model.plotTimescales command, which plots its timescales for you. And here we might choose a lag time, let's say, of 25 nanoseconds, where it's nice, flat, and also well separated, the 3 slow processes, the top 3 slow processes. So that means that since we have 3 slow timescales, we're going to pick 4 macrostates. 25 lag time in nanoseconds for macrostates, and that's it. That's how we build a Markov model. So let's load the model that I created before, so that we can continue executing stuff. So as I said, you can practically do the previous commands. It just take long, so I skipped doing them because I didn't want to spend time here looking at the screen. So now we can calculate the equilibrium distribution of all of our macrostates, the probability, sorry. As you can see here, the most populated macrostate is macrostate 3. Next one is macrostate 2, macrostate 1, etc. So, but what does that mean? What are these macrostates 4, 3, 2? So we call this view states command. Here, I just tell it to put visualization, a protein-based visualization. It's just for the representations of the viewer. But we might even take it out at some point to make it clearer. But so if we wait a bit, because it takes a bit to load the JavaScript NGL viewer.
So while we wait, which distribution are you using to sample from the macrostates? Something formulated in the macrostate?
No, I think right now we're sampling by the equilibrium probability of the microstates in the macrostate, but I think we could also sample by the
Output probability.
no, the membership probability of the microstates of the macrostates. Ah, you said output. Okay. Okay, so let's look at our states here. So it looks like a mess, but if you start disabling some states here, let's say macro 3 still looks quite messy, which is our most populated one, but it's actually the unfolded state, which makes sense because this protein actually is at melting temperature. But if you look at macro 2, it's actually our folded state, which looks very nice, which has its first helix here, second helix, third helix. So now if we go on to calculate the kinetics, we do it like this. We pass the model and we give a temperature. The temperature is needed so we can calculate the free energies. So it tells you that it's guessing the source state as a state with a minimum contacts. How much time do you have? You know, Johnny? So it finds that the state, it automatically detects what your source state is. For example, in protein ligand binding, it will find the one which has the least contacts or the largest distances. The same here, actually. It finds the one with the minimum contacts as being the most stretched conformation of the protein. That's your source state, and your sink state is automatically detected as the state which has the highest equilibrium probability, but which is not the source state. So if you remember the equilibrium probabilities before, the largest one was state 3, but state 3 is our source state. So the second largest one was state 2. So this one's going to choose as your sink state. Which is a nice heuristic. If you want to pass different source and sink states, you can pass them here in the constructor. There's options, source and sink. Source, sink, which you can pass. Also concentration if you're doing protein ligand binding. So then we can get the rates from the kinetics, and we can see the MFPT on, which is the mean first passage time of going from the source to the sink. In this case, that means that this is the folding time, and the folding time is around 1 microsecond. And the unfolding time is around 200 nanoseconds. As I said, the protein is simulated at a quite high temperature, and the free energy is also 0.8, which is quite close to what Dirshaw was getting, if I remember correct. If we want to look at the rates of all of our states, we can use this plot rates. We just create some plots with MFPT off, on, and free energy of each one of your macro states. Lastly, you can also look at the pathways. If you want to see how you go from state 3 to state 2, in this case it's extremely boring because all of your pathway pretty much goes from state 3 directly to state 2. But there is like a minor pathway going from state 3 to state 1 to state 2. The sizes here I think correspond to the equilibrium probabilities of the states in this plot. So yeah, it's nice for seeing the pathways. Okay. This concludes the part on the protein folding. I don't know if you have any questions on this. Otherwise, I'm going to go on with some stuff on ligand binding and some new methods.
Yeah, 30 minutes.
30 minutes, oh, okay. Maybe I was going too fast.
You probably have the time to conclude. without compute.
No, but I mean, yeah, you can also. Well, without compute. Okay, so now we're going to talk about protein ligand binding and some new changes that we've done, essentially. Well, not changes, some new features that we've added, even though they are partially experimental, still some of them, so we need to be a bit careful. But I'm going to talk first of all about how we are looking at protein ligand contacts. I'm going to talk about grouping selections for distances. Then I'm going to talk about splitting the bulk state. And lastly, I'm going to talk about core sets very briefly. So let's start with protein ligand contacts. So in the past, when we used to calculate the interactions between the ligand and a protein, we just looked at the contacts between, let's say, two atoms of the ligand here and the carbon alphas of the protein. But that's not really a nice proxy for interactions between the protein and the ligand, because the ligand doesn't usually interact with the carbon alphas, it interacts with the side chains of the protein. So this would be the selections that we use. So protein and name C would give us the carbon alphas, res name all is the ligand, and name C4, C6 would be these carbons on the ligand. So first of all, you have to manually select the atoms on the ligand, and second of all, we're losing the interactions with the side chains. But the good thing about it is that it's a low-dimensional representation, so you get like 2 times the number of residues of your protein as your dimensionality. On the other hand, now we are using kind of another selection, which is more automatical, which Gianni came up with, and also known as Gianni cell. So this selection essentially selects all of the oxygens, nitrogens of the ligand, and also all of the carbons which are not bonded to hydrogen. So as you can see in this ligand here, for example, this would amount to these atoms here. So this is a nice proxy because you can have strong interactions with those. And in the protein, we actually don't use just the carbon alphas, we use all of the atoms of the protein. So the problem with this is obviously that this is a super high dimensional representation, reduction. Well, not much of a reduction, let's say. So you have 5 times the number of atoms of the protein, which is very high dimensionals. But we can do one trick to reduce this to 5 times the number of residues in the protein, which would be, again, very trackable. And the way that you do it is by just grouping by residue your distances and keeping only the shortest distance between each residue and each atom of the ligand. So if you imagine this group here, for example, of the protein, these atoms, the shortest distance from all of those atoms to each one of those are these distances that you see here. So from this red atom, from this oxygen here to all of the other atoms. On the other hand, for this group here, you can see that the shortest distance for this atom does not correspond to the distance to this here, but instead to the distance to this atom. So we just keep the keep the shortest distance between the whole residue, which we've grouped into one entity, and each atom of the ligand. Exactly, and this reduces our dimensionality by a lot. So this can be done by using the option group cell residue in the metric distance class. So what would happen if we use group cell all? That puts our whole protein selection, like all of the heavy atoms of the protein, we put them all into one group, and then we'll just calculate the shortest distance between the whole group and each atom, as you can see here. So if we now group also the ligand atoms, then we're just calculating one single distance, which is the shortest distance between any atoms of those sets. This is quite nice because you can use this metric by grouping all and all. You can use this as a flag of if your ligand is interacting with the protein, which we are going to need for the interacting at all, like these flows to the protein, which we are going to need for doing our next step, which is splitting the bulk. So when we do analysis sometimes of GPCRs, typically we see it there, we have some states which kind of include both bound stuff, bound conformations, and also the bulk together. The reason for this is that we have, for example, a really slow transition inside here in the protein, But transitioning here is relatively fast. So if you do PCCA, so if we do our macrostates, we might end up with something like this. But if we want to calculate like the binding free energy, for example, we want to split the bulk into a separate state. We don't want it to be together with stuff that's attached to the protein. So what we do is we split it, as I said, the bulk into a new state, and now we can calculate the transitions from the bulk to a kind of attached state, from the bulk to the other one, etc. So we have a bit of a more detailed model.
You do this automatically, or?
I'm going to show it. Yeah, mostly. I mean, we can make it a bit more automatical, but essentially now we have one function where you tell it which microstates you want to split into a new macrostate, and just takes them and makes them a new macrostate. But I find no reason why not to make a function which automatically detects also the bulk. So, and the last thing that I'm going to talk about are corsets. So, okay, if we have like a two-dimensional potential here, I guess this is alanine dipeptides, I assume. And we have all of our microstates here, these small dots. I've colored them by the macrostate that they belong to. So as you see, we have like 3 basins here, for example. Once we do our macrostates, we see that they are kind of split with these barriers here, right? What Guillermo also mentioned before is that really we can have lots of fake transitions on the barriers between the states. So these, because the barriers are very arbitrary how we define those, stuff that's close to the barrier might transition back and forth very often and create fake transitions which make our model look like it's very fast, this slow process. So practically we are more interested in how long it takes to go from the core of this state to the core of another state than what's happening here on the barriers if we want to look at the slow dynamics. So the thing that we do in the corsets then is simply that we, well, there is some more detail, but you can imagine that we just take away the barrier, the states which are in the barriers, and we count the transitions between the cores. It's not exactly like that, but the details might change a bit because there are also different ways of calculating corsets. Right now we have a kind of fast implementation which uses PCCA, but there are other ways of doing it, so we might improve this also in the future. So yeah, this is the idea of the corsets, so that we only count transitions essentially between the cores of our states. So if we go now to the next.
The argument is that you don't need any more to have very refined discretization on top of the barriers, so you don't have any more the statistical problem that you had before that you increase a lot.
Yeah, the lag times that you can use decrease by a lot when you do corsets. No, I mean, it flattens much faster the times, because like even to a ridiculous point where it just starts flat. I've seen some times like that, which look kind of like a joke after you've done so many MSM analysis, but make you really happy.
The data has to fulfill some connectivity requirements for corsets to work.
Yeah.
You have to be sure that your data is usable with corsets, because otherwise you can introduce fake connectivity. But we can talk about this.
Okay, yeah. 1 second. Okay, so let's look at how we'll do a ligand binding analysis using our MSMs. Again, here we're not going to execute all of the commands because it was a huge data set, some 13 gigabytes, and we didn't want to put it on the EC2 server because it would take too long to move stuff around and we run out of space. So I'm going to talk about the distance selections that I told you before, splitting bulk, TICA dimension subselecting, which you need to do the bulk splitting nicely, and corsets. So for protein ligand contacts, as I said, it's more interesting to do something like this. So we define our protein selection, which are all of the heavy atoms of the protein, and our ligand selection, which is an S-name model for the name of the ligand, and oxygen or nitrogen or carbon not within 1.4 of hydrogen, which is this heuristic that we use for selecting interesting atoms on the ligand. And then you pass this to the metric distance here, for example, the prot cell and the lick cell. we ask for the contacts. We ask the contacts are considered the distances which are smaller than 5. And we also group the selection 1, which is the protein selection, by residue, which I showed to you before. So we calculate the shortest distance between each residue of the protein and any of these atoms of the ligand. We cannot execute this part yet. I will tell you again when we're going to start executing some of the code. So how do we create a bulk flag? So we first do, if we want, I mean, this is what I showed you before. This is the ligand selection that we use for the ligand, the protein. This is our first metric that I showed you before. So now we add another metric where we select all of the carbon alphas of the protein, for example. We select, again, the same atoms of the ligand, but in this case, we actually group both selections, both the protein and the ligand. We group into a single ball selection, group selection. So that's going to calculate a single number, which tells us if the ligand is within 10 angstrom of any carbon alphas of the protein. So actually, you could even not use carbon alphas here. You could use just protein and use all of the atoms of the protein. It would just take a bit longer to calculate, but not so much longer. I think it would be
It's really fast. It's all in C. And it's really fast.
Yeah. We've rewritten the distance calculation in C and even for groups, so it goes really fast.
It's actually faster to do the group selection, which seems counterintuitive, than doing the other one. Because
otherwise the other one. Debatable. So as you can see here, you can actually pass so the metric class that I was talking to you about, we'll pass it a sim list, but then we also pass it the projections that we want to use. And in this case, we can pass it a list of projections. And it's going to project on both of those. It's going to do both those projections and concatenate them into a single NumPy array. So this means that the data that will come out of this projection will have the interactions of the protein with the ligand, and as the last dimension, it's going to have this flag, the bulk flag, which will be 1 if it interacts with the protein, 0 if it doesn't interact with the protein. So we need to rescue our bulk metric from TICA, unfortunately. So when we use TICA, it normally takes all of our dimensions, all of the projected dimensions, and it projects them, and it calculates the TICA dimensions. So that would ruin essentially our flag that we just created for if the ligand is in contact with the protein. If we look at the mapping, so yesterday Joao showed you like these pandas maps, which give you a description of each dimension of your projection. If we look at them, the last one here, okay, it's not really intuitive right now, but it tells you that it's about the context of the molecule. It's because we cannot read the whole thing. It tells you that it's contact between the molecule and the protein. So it's a single number, and this is the flag. The last dimension is our flag. So how do we do this? Well, we go to TICA and we tell it to only project these dimensions, which are from 0 until your number of dimensions minus 1. So this is going to preserve our last dimension and not project it with TICA. And if you look at the data map afterwards, it looks like this. So the first dimension will be your flag, the flag that survived TICA, that we didn't pass to TICA. And the rest of the dimensions, which are the TICA dimensions, 1, 2, and 3. It even says here TICA, 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 and contact. So, okay, now we have done our TICA projection, and we've also kept our flag. So now we can do our k-means clustering. No, sorry, okay, we cannot do this because I haven't loaded the data. Normally you could. It's just that, as I said, for speed reasons, we're going to load here the cluster data directly. So this you can execute from here on. You can execute, I think, the cells. So we load just a model that I saved on the hard drive, and then we're going to plot the timescales for these lag times here, which I chose. Just
model.save, it's really useful.
Yes.
It saves an entire state practically goes in it because it contains the data, it contains everything.
Yeah, so if you're afraid that something is going to break or it takes really long to compute, you might just want to save the model, and then you're safe so that you can just load it back in and not have to compute everything again. So these timescales look a bit, so they don't really converge much. They take long to converge, and if you had to choose a point, well, I chose this point here, but it's I don't even know if Guillermo would agree. In any case, it's not a good model. We're just going to analyze it anyway so that I show you what improvements can be done on it. So I choose, let's say, a lifetime of 30 nanoseconds, 3 macrostates. The random seed that I put here, the only reason why I put this here is so that we could use less hard drive on the EC2 server, so I just kind of fixed our visualization to only use some specific trajectories. Because this command here, it samples randomly from your macrostates, conformations to visualize. And so that we didn't have to upload the whole data set, I fixed the random number generator. So it should visualize now our macrostates. Perfect. So if you look at this, there are essentially 3 states. There is the blue state, the red state, and the black state. The black state, as you can see, is both in interaction with the protein and not in interaction with the protein. It's also the bulk and also some stuff interacting with the protein. So we would like to split that actually. So if we look at the equilibrium distribution, most of the population is actually in that kind of bound-unbound state, in state 2. So if we calculate now, for example, the kinetics, we provide the temperature, we provide the concentration of the ligand, and we get a free energy, which is minus 1.78, which is not really that exciting. So let's try to do this separation of the bulk microstates into a new macrostate. So this code might not look very pretty right now. I'm going to explain it. We might make this a bit easier to use if we see that we use it a lot. Essentially, this command here is what does everything. So this create state, model.create state, will take the microstates that you give it, and it's going to create a new macrostate out of those. So the way that we are going to do it is by using the get state statistic function, which is quite useful, and you might want to try it out from time to time. You pass it a model, then you pass it a metric data object. In this case, I'm passing it the data that's inside the model, but it's not necessary. You can have like multiple projections of your data and pass it. So you might build your model with distances, for example, but then you might want to know in this state what is the lowest RMSD that I have. So then you're going to pass it here an RMSD metric. A metric RMSD object, so that it calculates the RMSDs of this model, even though it was constructed with distances. So here I tell you to give me back statistics for all of the microstates, and here I tell you to give me the minimum of all of the microstates of this data for each microstate. So this data is the TICA data that I showed you before. So it has as the first column, it has the bulk flag, if you are in contact or not with the protein. And then it has TICA 1, TICA 2, and TICA 3 dimensions. But we are only interested essentially in the bulk flag, which is the first dimension. So I just select the first dimension in this case. And I check whether the first dimension is equal to 0, which means that find all of the microstates which have at least a conformation which is not in contact with the protein. And if we print this, you're going to see that there are 3 microstates which are not in contact with the protein. And they all belong to the same macrostate, macrostate 2, which was our black one, as we saw before. With this here model.macro of micro, you can map microstates to macrostates. This is what this does here. I pass it the microstates and it maps them to macrostates, like this. Okay, so now that we split it, let's look again at our states. Takes a little bit. Again, this ligand selection here is just for visualization purposes. It's not. But should show its short thing. Okay, so now we have 4 macrostates. Okay. Okay, so if we look at it now, we have the blue state, the red state, our black state, and an orange one. It's not very clear now with the projector, but the orange one is a new state that we added. and it's essentially the one which is detached from the protein. The black one, it might look like this one is actually not in contact, but it is because there's this loop down here from the protein which extends a bit up in that conformation. So if you want, you can reduce the threshold of the interaction between the ligand and the protein, and then you're going to get an even finer splitting of the bulk. Okay, so now let's look at the equilibrium probabilities. Yeah, I think it's okay. Equilibrium probabilities. Okay, now we see that most of the probabilities in this black state, so the one that's like roughly attached to the protein, and we split out lots of its equilibrium into the bulk state, which is the second highest one in this case.
Which ones are in the bulk state?
The three. This is the bulk state. This is the one that's kind of attached, and the other ones which are inside the protein.
Sorry, the splitting of the bulk state was going from how many to how many? From 3 to how many?
4.
And
You add one new macrostate, which is the bulk.
The one that you split have how much? Like,
it had the most, yeah. That one that I split had the most one. So some part of it essentially went into the other one now. Yeah, into the bulk one. I mean, this model, as I said, is not really such a good model. I expect that the ligand is probably binding much stronger inside the protein, but I guess we don't have enough sampling yet. That's why also the timescales were not so good. But we're going to see what we can do essentially from this. So now if we look at the kinetics, we can see that now we actually get a minus 2.5 free energy, which is a bit better than the minus 1.7 that we were getting before. Okay, and last thing that I'm gonna show you are the core sets. They're still a bit in development, I guess. So this argument here, I'm not gonna talk about much. Just you can play around with it a bit until you get the flat timescales that you want. It's a bit arbitrary at the moment, but essentially this you call it from a model that we have created already at a specific lag time, and with some macrostates, and produces a new Markov model out of it, and a new model class actually. And then we plot the timescales, and you see that the timescales have become actually much nicer than before. Before they were kind of like here and not really becoming flat either. So here you could say that this is kind of a nice region where I would choose a lag time for the model. So around 10 to 15 nanoseconds, I think. Yeah, which is what I chose to use here. If we execute this, we get again the free energy, which is a bit boring. It's minus 1.8. But the thing is that once you create the core set, you're again ruining your bulk splitting that you had before. So if I do again the bulk splitting here, then we get a free energy, which is minus 2.28, which looks closer to what we had before. So yeah, this concludes, I think, everything that I had to say on Markov models, on core sets, bulk splitting, all of that. So if there are any questions, feel free to ask. Yeah.
I didn't quite understand the transfer definition of the Ligand key atoms. I think it was one sp3 carbon that actually would really accomplish this actually and you've got an aromatic ring now. We're always really biased. I don't know why, but when I look at medicinal chemists, stressing they do, oh, I've got a hydrogen interaction that's really important. But actually, you can look at aromatic interactions significantly, propagating interaction. So what I probably recommend is that you have a feature that's mapped onto, distal aromatic ring system.
Well, that's the idea with the carbon, which is another added. So the branching carbon. If you have one indicator that the aromatic group already.
It's just like it's not even so actually with aromatic ring you've got hydrogens on there, that's fine. But then you can map a picture of the centre of the aromatic ring.
But you don't have any atom in the centre there.
No,
But we did make one.
So at the moment you're missing quite a bit of aromatic ring, and the directionality that's really important. This is from a medicinal chemist perspective, that's usually very important, and usually the last sentence is in half it fits into the topic perfectly.
The problem with the directionality, that doesn't matter if it's like this or like that.
It can be
That's the same thing. Like this or like that.
Got by stacking interaction.
No, I mean, it doesn't matter if the ring is like this or this is 180 degrees.
But I want to know it's going into the pocket though. So I need to have some feature that's mapping on there. So from some,
You can, because if you have something that's mapped that carbon, you are artificially splitting that. So the macro model will see sometimes going like this being stable and sometimes being like this being stable, and it doesn't recognize that it's the same thing. Because
but if you create a dummy atom for the ring,
if you get dynamic, the center is 1 angstrom away downward.
I just want to do that just to get a feature because actually there's no point in that carbon that you map next to aromatic ring. It's not telling you anything.
Sorry?
I'd rather go, you know, an angstrom or so away and just try and get more information in the center of that aromatic ring that have that carbon that's in the center of that. That's not really
But use threshold of 6. But
yeah, but
it's not that important where you put that. It's not 1 angstrom away important.
well, it depends on how fine you want to do the interactions that you
That's
resolve.
what is important.
I think if I might interject, I think you're speaking about slightly different things. What Jan is saying is, whatever the actual interaction is taking place, you're looking for a proxy, a geometrical proxy that will map that interaction is taking place. So you don't really necessarily have to be mapping features of the atoms responsible for the interaction, just any proxy.
But that proxy might not tell me that interaction.
Depends on the threshold that you use for contacts. That's what Johnny was saying essentially. That if you use a larger threshold for the contact, then okay, it's gonna be in contact even if it's there on the ring, but you might lose some details of your interaction, I guess. Yeah, depends on how much detail you're willing to lose in that case. No?
I mean, this selection, the only use of this selection is to reduce the minimum. The only use of this selection is it's very simple to write, and it reduces to bare minimum the number of atoms. Because TICA is quadratic in the number of things. If you use all the atoms there instead of 5, it goes maybe it's 10, I don't know, without the adjectives. It will go 4 times lower. If it's 3 times, it goes 9 times lower. So it's between having a TICA of 20 minutes and a TICA of 200 minutes.
Yeah,
that's the only reason.
Yeah,
There is
I
actually
appreciate that. And enough methods that we use in industry and to just map the atoms onto aromatic rings. It could be a 9-membered ring actually. There's a lot of nanocycles that are interesting these days. So there's a lot of things where once we do dimensionality, there's some feel for whether it's interacting. And maybe if you can introduce the atom option when you can map it.
Yeah, I mean, if we put dummy atoms, then we can model interactions better, I guess. Right now, we don't have the functionality for that, but
Would it be possible to put together like a smart definition you can feed in and say I want that sequence cube model reactor?
smart definition? Well, I can imagine, yeah.
So you can go just with the center of mass.
The problem is that for each frame, you must recompute essentially your dummy atoms. So because the ligand moves around, you need for each frame of your simulation to recompute your dummy atoms.
You can do it manually if you want. But for the ring, you can select the ring. I don't know if it's a selection.
Group it, yeah. Well,
and then do the minimum distance of that ring.
practically minimum distance, I think, are more interesting than dummy atoms, essentially, because they give you the closest interaction, which is yeah.
That's the way to go if you want to really be specific. You can break up your business different metric and each metric just to do it separately. This is just a very good way of giving a reasonable small number.
Yeah, I completely appreciate the dimensionality.
I mean, as a comment of what Diego mentioned, how expensive is to measure the center of mass?
To measure it.
Not to measure, like to use the center of mass instead of like.
Not much.
But why not doing it?
As long as you don't.
No, right now you mean like using dummy atoms. I haven't listened to that. Doing the grouping. No, it's nothing. It's
Because we don't care.
but actually, what of the whole ligand, the center of mass of the whole ligand would be less important. Because then you need to do actually separate selections. You could. Then you make one selection for the aromatic ring and one for the rest of the ligand. But the problem with that is that you need to. The nice thing about this is that it's mathematical. So you could use a selection of different ligands. In the other case, you need to look at your ligand and say, okay, there is an aromatic ring here, make another extra selection for that aromatic ring. I mean, okay, if we have some detection of aromatic rings, we could make some groupings and stuff like that.
right?
Yeah, I think the idea is just that it's a simple selection, which kind of works well. Yeah.
A small question: is it possible to include the analysis besides the number of the contacts, the clusterization of the established interfaces? It's more important for the protein peptide binding because peptide, which is a ligand, can rotate and to make different poses inside the receptor, providing at the same time the same contacts. So we can to make the
Well, it won't have the same contacts as long as it's.
make
How do you mean that the whole pocket moves
yes,
with it?
how the ligand rotates inside the pockets and to which contacts it results. Because I worked in the same project with protein interaction.
You mean a case like where the ligand is inside the pocket and then they both rotate, for example?
No, the ligand, it can.
Well, if only the ligand rotates and depends on your selections on the ligand, it might produce different contacts and on the threshold of your contacts.
Yes, yes.
I mean, if the contacts are really short and you're doing grouping by residue, so you're getting the shortest distance to each residue, then you're going to get different contacts depending on the orientation of the ligand, I guess.
So the selection of the threshold is most important.
Yeah, I mean, okay, if you use a large threshold, you're making everything kind of a soup. You're always in contact with everything. So, yeah, I would say that if you want to separate those. Actually, another interesting subject is about symmetrical poses of the ligands, but that's not easy to handle. Like in mezamidine, for example, it doesn't matter if it binds like this or if it binds like that in the pocket. You want to consider them probably the same. So there are ways of doing it manually. Yeah, I don't know. Yes.
If you've got the fluxes as you did before, in the first model, you get that the main flux is from the bulk state to state, so from 2 to
from 3 to 2.
From 3 to 2. But when you split in the next model the bulk state into 2 states.
You meant here in the ligand binding.
Yeah, in the ligand.
Okay. I don't know what the fluxes look like. Yeah.
So yeah, let me check. In one case, you have from 2 to 1, which is bulk to one of the positions.
Yeah.
Inside, which makes sense. And then the state 0 is not connected. But if you split the state 2 as you did,
I know. Yeah.
then the flux is completely changed.
Yeah, I know the reason for this. It actually makes sense because the sink definition changed in the second model. If you look at what the sink was in the first model and in the second model, it changed. In the first model, we had like 3 states essentially.
Yeah.
No, we had 3. Yeah. Yeah, but 2 of them were bound inside the protein, like very inside, and 1 was kind of this mixture. So this mixture was determined as a source. So the sink has to be something other than the source. So it picked one of those which are inside the protein. Once I split the bulk, the source becomes the bulk, and the sink becomes the one that's barely interacting with the protein, like kind of interacting with the protein. So yes, you're going to get just by default, you're going to get just the fluxes to go from the bulk to this barely attached state.
Yeah.
But if you want to actually see what the fluxes are to go to one of the inner states, just change the sink to one of the inner states. The reason why it's chosen as a sink is because it has a really high equilibrium probability, this state. So practically the model says that these ones which are inside the protein, we don't really care about much because they are not very probable. The problem is probably the model. We don't have enough sampling. I assume that it's better, that it has a high equilibrium probability to really bind inside the protein, but since we don't have enough sampling, it doesn't find that. So that's the reason why you get the different fluxes.
So to compare them, you should specifically use the same thing.
Yeah. Also the kinetics that I actually calculated after I split the bulk, they are not practically the same, the free energy that I calculated. It's not between the same states anymore. So now I change the states where I calculated. So if I really wanted to compare it correctly, I should choose the other one as a sink state.
In a convergent model where that thing is bound for the
yeah, exactly.
Heuristic would be correct.
Yeah.
That's why we print it, because it's not always correct. There's no way to make it always correct.
Yeah, that's why we print out that it goes between this state and that state, so that you can actually check if it makes sense for you which is the source and which is the sink. And yeah, we have to be careful with that, especially when we look at the flux pathways.
Another question? Okay, so a copy break, however,